Hello and welcome to our annual ICRS Ethan Russo Trainee Prize fundraiser and webinar. We're so happy that you guys have had the time to join us. We're very excited about the presentations that we will have this hour, and I think you're going to have lots of questions. So just a few housekeeping details. Please put your questions in the Q&A box, which should be at the bottom of your screen. You guys are welcome to chit chat and make comments in the chat box, but we're using the Q&A exclusively for questions that we want to ask of our presenters. So the way this will work today is I'm going to talk briefly about the prize itself, and then our honoree will introduce the two most recent winners of the prize, and then they will each present, and after their presentations we'll take questions from the audience. So we are here today to raise money for the ICRS Ethan Russo Trainee Prize, which is really to motivate and to honor young scientists that are asking the big, bold questions that ultimately will translate into medical practice. And as most of you probably already know, funding for research is limited and hard to come by. And so we have this community funded scholarship that was created 5 years ago in Ethan's honor to recognize the incredible contribution that he has made to the area of cannabis science and cannabis medicine. And without pioneers like Ethan and other wonderful colleagues such as Beth and Radka, we aren't able to move this field of medicine forward. And so that's why we're asking for your contribution, whether you have $5, 50, 500, 5,000 or more to contribute. Every dollar helps. And over time, it is our goal to significantly increase the amount of prize money that we can give to a deserving trainee. But we can't do it on our own. We need your help. And so many ways you can help. If a donation is not in the cards for you today, you can also share the trainee prize information across your social media networks and to talk about how important it is that we all support the research, because that's the only way that we get to move this field of medicine forward. So with that, I'd like to turn it over to my colleague and my dear friend and my business partner, Dr. Ethan Russo, who is our honoree today, to say a few words and introduce our phenomenal speakers. Ethan?
Thanks so much, Nisha. Hello, everyone. I'm going to keep this very brief because we're all really interested in what these young scientists have to say. So we'll hear first today from Beth Wiese, who will talk to us about use of cannabidiol to help prevent respiratory depression associated with opioid overdoses. Beth is a PhD and had postdoctoral training, and I was really struck by her presentation. She was the awardee in 2024. So we'll be hearing from her first, and then followed by Radka Kocvarova, who's coming to us from Israel. As you will see from her name, she is Czech. Radka has really been very brave to join us. She's experiencing an unwanted event, having to go to shelters because of air raids currently underway in Israel. So we'll be using a recording, but hopefully Radka will be able to stay with us and do the live Q&A. So with that, Beth, could you go ahead and please start your presentation? All right, Beth, take it away.
All right, thank you so much. Yes, one more, there we go. Hello, everyone. My name is Dr. Beth Wiese. I'm going to talk to you today about the emerging role of CBD in overdose prevention. And we'll share the slides afterwards, so you'll have my bio. And I always like to take a moment to make a land and labor acknowledgement and recognize our location on the ancestral land of, I'm in Los Angeles, the Tongova, Tativam, Serrano, Quiche, and Chumash peoples, acknowledging their ongoing stewardship and the harms of settler colonization. This acknowledgement honors the elders and the descendants of these tribes, recognizing their ongoing stewardship of the land and waters, acknowledges the harms caused by settler colonization, including land seizure, disease, subjugation, and other injustices. This acknowledgement is a commitment to truth, healing, and reconciliation with the First Peoples. And we acknowledge that our nation has benefited from the free and slave labor of Black people and people of color. We recognize the forced labor and debt owed to Indigenous peoples, Chinese immigrants, and Japanese Americans for their contributions and the exploitation that they faced. We acknowledge and remember those who did not survive the Middle Passage, those who were beaten and lynched, and those who are still suffering while fighting for their freedom. We honor the legacy, knowledge, skills, and human spirit that persevere despite violence and white supremacy. And this work would not be possible without the prior contributions of immigrants. And with that, the continued problem. In the US, at least, and around the world, we have experienced an overdose epidemic that has been on the rise for multiple decades now. And in 2023, it got severe enough to have over 200 people a day who died of an opioid overdose in the United States. And while these numbers have started to decline slightly, we are not out of the woods yet. This is still a major problem, and it is a preventable death. And so we know that an opioid overdose does not have to be fatal because we have a medication that is able to restore breathing. And so when someone stops breathing from an opioid overdose, what happens is the opioids inhibit their reflexive breathing. And then we have a medication called naloxone that can be given to restore breathing. That naloxone is an opioid antagonist. And the way that it works is when there's an opioid on the opioid receptor and naloxone comes in, it comes in and kicks that opioid right off the receptor and allows breathing to start again. But the problem with naloxone is it doesn't just block or it doesn't just restore breathing. It also blocks pain relief. It induces withdrawal in people who are opioid dependent. And it can only be used after breathing stops because it cancels out all the other effects of naloxone. And so naloxone is really vital to be able to have in the hands of people who use drugs. And just to speak to how powerful it can be, in Los Angeles, I work with a harm reduction organization that is based in Skid Row. And we have what's called the Tree of Life program that meets the community where they are. And so we go up and down the streets, and peers will go up and down the streets, and they staple naloxone to the trees and the telephone poles. So that way that's always available. And people know that when something happens, that if there's no one around, they can look for naloxone to be present on the trees and the light poles. And so in 2025 alone, the Sidewalk Project distributed almost 40,000 naloxone doses that resulted in over 668 successful reversals. And this speaks to how powerful it is to have life-saving medications in the hands of the people who need it the most, people who are using drugs. Naloxone is also one of the easiest drugs to train people on how to use, especially the ones that are shown here. These are all nasal. And so we can teach people who use drugs how to recognize an overdose and then how to use the nasal naloxone to restore breathing and save someone's life. And in addition to the naloxone itself, we have an overdose response team. And so what that means is that in Skid Row, when someone does experience an overdose and peers begin the revival process, the reversal process, someone else can also come and get us from our center as we are located in Skid Row. And once we're mobilized, we take the cart that you can see in the middle, and along with other overdose reversal items, and we run to the scene. And so not only do we administer naloxone, we utilize this belt sensor that is in the upper right-hand corner, and it's a stretch sensor that lets us know immediately when someone starts breathing again. It Bluetooths to our computer, which you can see attached to the cart in the middle that we take with us into the streets. And the video down to the lower right is a kind of up close picture of what it looks like when somebody is wearing the belt and they're breathing. And so this lets us know immediately when we can stop giving naloxone because breathing has begun again, and we can move to just oxygen administration. But what if there was something that we could do to prevent overdose before it occurred? So during my time at UCLA and my postdoc, I was doing preclinical research and I was looking at fentanyl administration to awake behaving mice. And so I would place these mice into a whole body plasmography chamber to measure pressure changes as they breathed. And I would pre-treat the animal with either saline or vehicle in this situation. And then I administered 50 mg per kg of fentanyl, which decreased the breathing in mice by approximately 50%. Next, I pre-treated with naloxone, and then I administered the 50 mg per kg of fentanyl to the mice. And the naloxone was able to prevent the respiratory depression from the fentanyl, which is to be expected. What was exciting was when I pre-treated with CBD, a high dose of CBD equal to that of Epidiolex. I was also able to prevent the respiratory depression that was seen from fentanyl with just a saline or a vehicle pre-treatment. And then finally, I tried a pre-treatment of CBD and naloxone to try and investigate synergy. But in awake mice, it is much harder to see that from this model that I was using. It also did prevent the respiratory depression from the fentanyl, but it was not possible to really tell if one or the other, or if the combination of naloxone and CBD were what was causing this prevention of respiratory depression. And so since I wanted to really get at whether or not CBD could be used as a preventative to overdose, and it's very difficult to administer a fatal dose of fentanyl to a mouse in time to get them into the whole body plasmography chambers and measure that, I had to use a different model. And so that model is a model of overdose. And so in this model, what I did is I anesthetized the mice with urethane anesthesia, which is often used in sleep studies because it mimics awake breathing behavior while the mice are anesthetized. And so with that maintained breathing behavior, I could then start a fentanyl intracranial ventricular infusion. So a continuous infusion of fentanyl into the brain at 100, or into the ventricle at 100 nanograms per minute. And I could evaluate what was happening to the breathing behavior all the way to the point of when the animal would succumb to an overdose. And you can see here how breathing behavior would changes over time during this overdose. And so again, I did pre-treatments. Once the animal was under urethane anesthesia, and I would give a pre-treatment of saline or vehicle, and then I would wait 10 minutes and start the fentanyl infusion. And with the anesthetized model in this model of overdose, the animals pre-treated with saline or vehicle succumbed to a fatal apnea within 9 minutes. And when I pre-treated animals with naloxone, it delayed the apnea onset almost double to about 18 minutes. And then when I pre-treated with CBD, it was again equally as effective as the naloxone alone to delay the apnea onset. And when I gave the two medications together, CBD and naloxone, as the pretreatment, I saw a further delay in apnea onset and a prolonged survival time. And so, where the individual compounds, naloxone or CBD, were able to prolong the apnea onset to approximately 18 minutes. When I gave them together, it prolonged the onset of the apnea to over 30 minutes, which suggests that there's a potential there for increasing the safety profile of opioid therapies by utilizing CBD as a preventative. But this was in mice, right? And so we can't just move straight into the field with it. And so, will it translate, and how would we test this given the sensitive population that we're working with? People who are dependent on opioids, generally illicit opioids. And how would we investigate whether or not CBD could be effective? Well, during the fires in Los Angeles last January, we had to open our shelter in Skid Row as a 24-hour shelter for people escaping the fire and being evacuated. And during that time, the community really stepped up and came together with donations and support and looked out for each other. And one of those big donations that we ended up receiving came from a local dispensary that provided us metered CBD vapes. And so while this is not a clinical trial, but we've got these belts that are able to measure respirations through a stretch sensor, and we had this bulk supply of CBD vapes. And so when we were talking about this, we decided we really needed to partner with an institution or an academic institution to help promote this research. And so you're looking right here at my desk at the Sidewalk Project in Skid Row, where I started a research institute called Thrive, or Transformative Harm Reduction Research Institute of the Voices. And this is the institute that we are using to continue this research and investigate how we can move these results forward into and translate them into real world interventions. And so, with the Sidewalk Project being a harm reduction organization, pretty much everyone who works there is someone with lived experience of either using substances or someone who is currently using substances. And we have several staff members who do currently use opioids for pain management. And so specifically what we did is working with one of our staff members who takes hydrocodone to manage pain. They would come in and we would collect a breathing baseline for 10 minutes. and then they would take their pain medication when they would always take their pain medication. So this observational timeline did not interfere with how they normally take their pain medication or dose themselves. And so after they would take their hydrocodone, we would wait 30 to 60 minutes and collect another reading, another breathing recording, a breathing behavior recording. And the reason that there is such um, a difference, open area, a long time for this recording, 30 to 60 minutes, is because this is a staff member who is also still carrying out their work duties at the same time. And so we didn't want to interfere with anything. It was when, they were able to come back for another recording. And then after that recording, they would have one of the CBD vapes that they could puff as many times as they wanted. There was no limit or restriction, just to let us know how many puffs that they had taken. And it was between 2 and 5 each time that they would use the vape. And so that's 20 to 50 milligrams of CBD. And then after that, we would collect another 10-minute recording. And so, oops. And so these are the preliminary observational findings. So we would record baseline, and then after taking hydrocodone, you can see that the breathing rate would depress to about 80% of the baseline rate. And then after vaping the CBD, the breathing behavior would return back to baseline, and sometimes even higher. And that was on 20 to 50 milligrams of vaped CBD that restored hydrocodone-induced respiratory depression in an observational timeline in humans. And so with this information, the future directions of Thrive that we are looking for, or looking to continue with, is to partner with new organizations to create collaborations where we can investigate this, whether it's potentially in Canada alongside legalized safe consumption sites where people can be observed while utilizing CBD and their substances. We are working on grant writing and financial support to support these translational studies that could have real world impact. We're hosting community involvement and feedback sessions, or community advisory boards, where we have participants from the Sidewalk Project who are able to come in and provide us with the questions that are of their interest for the Research Institute to be investigating. And then we are working on program expansion and ways that we can build out the BELT program and the for overdose response, and for future experiments and findings. And with that, I want to say thank you and acknowledge some really wonderful people who have been able to assist in making this work come to life. And that includes the funding from the Robert Wood Johnson Foundation, my colleagues at the Sidewalk Project, the Los Angeles Fire Department and EMS, the AIDS Coordinator's Office in LA, who funded the initial pilot project, Mockingbird Analytics, the Department of Public Health and Substance Abuse Prevention and Control, or SAPC, in Los Angeles, and overdose response teams in Skid Row, as well as Dr. Russo and Nishi for having me here today to present. Thank you very much.
Thank you, Beth. That was lovely. All right, so now we will go to Radka's presentation.
Hello, everyone. I'm delighted to be part of this wonderful event. My name is Radka Kocvarova. Well, I'm originally from the Czech Republic. I've spent the last 5 years pursuing my doctoral research at the Obesity and Metabolism Laboratory of the Hebrew University of Jerusalem, under the supervision of Professor Yossi Tam. I'm really excited to share with you today the highlights of my work on phytocannabinoids for treating obesity-related fatty liver disease. Given my 20-minute time budget, I'll provide more of a high-level overview. So for those interested in these specifics, I will share our recent publication at the conclusion of the talk, and please feel free to contact me with any questions. So let's look at the bigger picture first. Obesity is something we all hear about, but the scale of the problem is far more alarming than most people realize. Obesity is a chronic progressive disease that has reached epidemic proportions globally. Today, 43% of adults are overweight. That means nearly half of the world adult population is affected. And obesity is not just about excess weight. It has severe metabolic consequences such as cardiovascular disease, type 2 diabetes, and metabolic dysfunction associated steatotic liver disease, or MASLD in short. The letter 1, MASLD, refers to a branch of conditions caused by a fat buildup in the liver, which is unrelated to alcohol or viral causes. It affects about 3/4 of all obese individuals. Given how common obesity has become, it's not surprising that the rate of MASLD has also increased dramatically. Without intervention, MASLD can progress to MASH, cirrhosis, and hepatocellular carcinoma. Even at these advanced stages, an individual might have no symptoms or only vague ones like fatigue or nausea. The good news is that steatosis is reversible if detected early. However, there are no currently approved drugs for treating it. Evidence recently emerged that some drugs that are already on the market for other conditions can also ease MASLD. A good example are the GLP-1 agonists, primarily used to treat type 2 diabetes and obesity. But they also show improvements in steatosis. While this is promising, they're not registered for MASLD and can't be prescribed for it. Another milestone is resmetirom, approved as the first drug for MASH. While resmetirom does reduce liver fat to some extent, it is mainly aimed at the fibrotic scarring that is usually not present in muscles, but only in the later stages like MASH or cirrhosis. Therefore, the unmet need remains for a specific effective drug for MASLD. Our lab, based at the Hebrew University of Jerusalem, has long been researching one of the body's key metabolic regulators, the endocannabinoid system. It is a ubiquitous signaling system found throughout the brain and the body. It acts as a crucial metabolic regulator, controlling functions like appetite, energy balance, and fat storage. The system consists of three main components. We have the endocannabinoids. These are ligands produced naturally by the body, such as 2-arachidonylglycerol and anandamide. Then there are receptors, primarily cannabinoid receptor type 1 and type 2, which receive the signals. And lastly, enzymes that synthesize and degrade the endocannabinoids to control their signaling activity. The endocannabinoid system can also be influenced by compounds that come from outside the body, specifically from the cannabis plant. Phytocannabinoids are natural compounds produced by this plant. There are more than 150 known phytocannabinoids. Each of them has distinct biological effects. They interact with the endocannabinoid system to a different extent, while also acting on other molecular targets. Examples of phytocannabinoids include THC, known for its psychoactive effects, as well as non-psychoactive compounds such as CBD, or cannabidiol, and CBG, cannabigerol. Around 10 years ago, key papers showed that even a weak CB1 receptor ligand, like CBD, could effectively reverse hepatic steatosis. This was an exciting finding, but it also raised a fundamental question. How do these compounds actually work on metabolic disorders? It can be only through the endocannabinoid system, since CBD specifically has very little activity there. This knowledge gap is exactly what prompted our research work, which we have structured into two main objectives. Number one, characterizing the metabolic effects of CBD and CBG in diet-induced obesity model. And number two, deciphering the molecular mechanisms of their action. So let's begin with objective number one, characterizing the metabolic phenotype. So for our study, we used male C57 black mice that had unrestricted access to food. Mice on standard diet served as healthy controls, and other groups were fed high-fat diet for 14 weeks to induce obesity and fatty liver. Following this, mice were injected daily for 28 days. Control groups were given a vehicle, while experimental groups received CBD or CBG. During the treatment period, at no point did the phytocannabinoids affect body weight. However, despite weight not being affected per se, we did find a significant reduction in fat mass and an increase in lean body mass in the CBG-treated animals shown in purple. So we asked, could differences in food intake or activity explain these results? Surprisingly, mice from all the groups actually ate about the same amount of kilocalories a day, and the CBG group even moved less than the high-fat diet controls. Pedestrian locomotion, ambulatory activity, and wheel running were all lower or unchanged. Yet, these animals showed improved lean-to-fat mass ratio. This suggests a shift in metabolism rather than just changes in energy balance and behavior. Next, we explore how this metabolic shift translates to metabolic health markers, and we observe some very clear benefits. Both CBD and CBG treatments normalized high-fat diet-induced hyperglycemia. Next, we use the glucose tolerance test to assess the body's ability to handle a glucose load. By measuring the area under the curve, we confirmed that the ability to utilize a bolus of glucose is clearly normalized with the phytocannabinoids. Next, we evaluated insulin sensitivity using the HOMA-IR, which is an index for quantifying insulin resistance and beta cell function. We found that CBG significantly lowered HOMA-IR, showing a profound improvement in insulin sensitivity. Beyond glucose and insulin, we investigated the impact of phytocannabinoids on dyslipidemia. Remarkably, both CBD and CBG dramatically reduced serum triglycerides, actually bringing them below the levels seen in our healthy control group. We saw a similar restorative effect on total cholesterol, driven primarily by a significant reduction in LDL. But the most striking findings emerged when we examined the liver for steatosis. We measured steatosis in two ways. First, quantitatively. We found overall hepatic triglycerides and cholesterol drop dramatically with both CBD and CBG. And then we looked morphologically. The upper panel shows H&E stained liver tissue. We can see here fewer fat vacuoles and balloon cells in the treated mice. The lower panel contains all red O stain to dye lipids in red. Quantitatively, high-fat animals had about 15% of the fat, but the treated groups were sharply reduced to only about 1 to 5%. This slide summarizes the key findings from our phenotypical characterization. It confirms that CBD and CBG can, in diet-induced obese mice, successfully reverse hepatic steatosis, improve the lean-to-fat mass ratio, normalize glucose homeostasis, improve insulin sensitivity and beta cell function, and also resolve key dyslipidemia markers. All these metabolic improvements were independent of changes in food intake or activity. Now, objective 2 is dedicated to achieving an understanding of how these compounds function at the molecular level. So, building on the endocannabinoid system importance we established together earlier, we went into objective 2 asking, do CBD and CBG modulate the endocannabinoid system signaling at all? First, we measured the levels of endocannabinoids and endocannabinoid-like molecules. These are fatty acid derivatives, similar in structure to endocannabinoids, that help regulate the endocannabinoid system activity. Both these groups remained largely unchanged after the treatment. There was an increase in anandamide only with CBG. Similarly, gene expression of the endocannabinoid system-related genes was unchanged. Overall, these data suggest that CBD and CBG work through a different endocannabinoid system-independent pathway in our setting. So before we chase the mechanism further, let's remind ourselves how the liver normally handles lipids. There are two main routes how fat gets in. Number one, here direct uptake of fatty acids from the blood. And number two, lipogenesis, where the liver makes new fat from excess carbohydrates. And on the other hand, there are two main ways how to get rid of fat from the liver. As number three, beta oxidation, where mitochondria burn fat for energy. And number four, VLDL export, which packages fat into lipoprotein particles and sends it back into circulation. In obesity, this usual equilibrium is lost and fat accumulates. So our next step was to test whether CBD and CBG could restore this balance. First, we looked at the fatty acid influx. We saw downregulation of some fatty acid transporters at the gene level. However, when we looked at the proteins, specifically CD36, which is a major fatty acid transporter, we didn't observe a matching decrease. So while fatty acid influx might be slightly modulated by CBD or CBG, it likely does not explain the full extent of liver fat reduction. Next, we quantify key lipogenesis genes. Some changes were detected, but overall the effects of CBD and CBG on this pathway were modest and likely not sufficient to explain the improvements in MASLT. We also tested fat oxidation directly in metabolic chambers by measuring carnitines, because carnitines are essential for transporting fatty acids into the mitochondria for oxidation. We did that using metabolomic profiling and also by measuring related genes. When you look at the graphs, you see that fatty acid oxidation actually decreased with CBD and CBG, which is the opposite of what we would expect if extra fat burning were behind the fat reduction. So these first three mechanisms turned out not to be the answer. We had really high hopes for the last pathway, fat export through VLDL. The results, however, were disappointing. We used FPLC to measure VLDL directly, and we also analyzed triglyceride content in the lipoprotein fractions. There was no change at all, so fat wasn't simply exported more either. We had a mystery. MASLT was clearly reversed, but none of the usual pathways could fully explain it. That's when we turned to omics, metabolomics, and lipidomics to get an unbiased view of what was happening to the liver's metabolome and lipidome. Let me start with metabolomics. We completed large-scale metabolomic profiling of the liver using LCMS. We screened for over 500 metabolites, of which 367 were detected in these samples. Almost 1/3 of them significantly changed with at least one of the treatments. But what really stood out was the large increase in creatine and phosphocreatine levels with both CBD and CBG treatments. Phosphocreatine is a high-energy molecule. When energy is required, it donates a phosphate group to ADP, converting it into ATP. This ATP then provides energy where needed. Creatine kinase is a bilateral enzyme that catalyzes this conversion. While this system is especially important in tissues with quick high energy demands like the muscles, it is present in almost every tissue. I'm showing here several articles that link creatine supplementation to the improvements of liver steatosis. However, to the best of my knowledge, there is no information in the literature about phytocannabinoids reversing steatosis through the creatine phosphocreatine system. So next we investigated the system key enzyme, creatine kinase. In the liver, we found a significant upregulation of the creatine kinase variant 1 gene. in the CBG group, with a positive trend in the CBD group also. Variant 2 was undetectable here, which aligns with its known tissue-specific profile. We also checked the kidneys, as creatine metabolism is shared between these two organs. In the kidney, however, both variants were either lower or unchanged. Additionally, we measured creatine kinase activity using a UV-based assay. In the liver, we found a significant increase in activity with the treatments, which directly correlates with our gene expression results. Conversely, in the serum and muscle, which are the primary sites for the creatine energy system, we observed lower activity compared to both control groups. This points to a liver-specific effect. So let me summarize this part. CBD and CBG-induced increasing creatine phosphocreatine system gives energy support to the liver, while fatty acid oxidation and other usual energy-yielding pathways remain unchanged or even lower. This suggests a potential mechanism for CBD and CBG in reducing hepatic steatosis. So after I showed you the metabolomics result, let me follow with lipidomics that helped us understand how CBD and CBG impacted the liver lipidome. We ran a lipidomic analysis of the liver that identified more than 2,000 lipids across multiple lipid classes. This heat map revealed distinct clustering of treated versus untreated groups. We saw three patterns. In response to the treatment, the lipids in the top cluster shifted toward the healthy, lean standard diet profile, while those in the middle remained similar to high-fat diet group. However, the lipids in the bottom cluster increased, diverging from both standard diet and high-fat diet profiles. Next, we grouped the lipids by class. These pie charts show classes of lipids that were significantly decreased. We see mainly triglycerides and ceramides. Since triglyceride accumulation is a hallmark of steatosis and ceramides are linked to insulin resistance, the reduction is a positive metabolic adaptation. Other lipids were increased in response to the treatment. Among these, we see LBPAs and several phospholipids. LBPAs are crucial for the endosome-lysosome system, and I will speak about it more in detail in the next slide. And phospholipids are key for maintaining healthy cell membranes, among other functions. So now let's focus on the LBPAs. These lipids support structure and function of late endosomes and lysosomes. Lysosomes digest many cellular components, including protein, sugars, and also lipids. In muscle and obesity, lysosomal function is commonly impaired and LBPA levels drop. But with CBD and CBG treatment, LBPA levels highly increased, suggesting improved lysosomal function, which could help the liver work better and clear excess fat. Inside lysosomes, we find catepsins, enzymes that break down cellular material. As shown in the top graph, total catepsin activity is a composite of three key subtypes, B, S, and L. In our high-fat diet mice, this activity was significantly suppressed, a clear indication of lysosomal dysfunction. But after CBD and CBG treatment, the activity was restored, in some cases even above the baseline. So not only we do see more of these structural lipids that support lysosomes like LBPAs, but also signs that the internal enzymatic machinery is back in action. And it's not just the activity. Looking at gene expression, we found that catepsins B and S were significantly upregulated in response to both phytocannabinoids, with catepsin L also showing a strong upward trend. This says that the late endosome lysosome system is transcriptionally activated. So to summarize this objective, CBD and CBG restored lysosomal function, which is commonly impaired in muscle. We saw increases in both LBPA and catepsin activity. So we are looking at a novel mechanism by which CBD and CBG may reverse fatty liver. So what is the main takeaway from this research? Ultimately, we observed a significant metabolic restoration where CBD and CBG treatment effectively reverse fatty liver in our diet-induced obesity model. We found this works through two main pathways. First, provide an energetic support by activating the creatine phosphocreatine buffering system, which helps cells maintain their energy balance under metabolic stress. And second, they restore lysosomal function, restarting the essential enzymatic machinery and the cell's natural waste disposal. We truly hope this research lays the groundwork for an effective treatment, because for people with fatty liver disease, those medical options simply do not exist yet. I'm very happy to share that the work we've discussed today was published in the British Journal of Pharmacology less than a week ago. And of course, a project like this is never a one-man show. This research was a true team effort, and I want to thank all the amazing people whose names you see here on the slide. I'd specifically like to point out my mentor, Professor Yossi Tam, whose guidance and support were absolutely vital. And finally, I want to take a moment to thank Dr. Ethan Russo. Winning the Trainee Prize last year was a huge honor, and your support for early career researchers like myself is exactly what allows this kind of science to move forward. Thank you so much, and thank you all so much for listening.
Well, those are two outstanding presentations. Thank you both. So, Radka, I'm going to offer the first question to you because I know you may have to go here at any moment. So, you mentioned that your goal with this research is to get it into clinic. So, can you elaborate about that? Do you intend this to become an over-the-counter treatment or a pharmaceutical drug? What's your hope?
So this part of work, thank you for the question. We will not do ourselves as we focus on the preclinical studies. We'll probably move it on to experts that do clinical studies. But the hope is to get it to the clinical practice as a prescription drug, to have at least some options other than lifestyle changes for people with fatty liver disease.
Thank you. And so our next question is for both of you. And given the situation you're in, I'm going to ask you to comment first, and then Dr. Wiese will have you answer. So what were the challenges that each of you faced in conducting this research, and how did you overcome them?
Radka?
So I think my biggest challenge was that first I use an targeted approach. As you saw in the presentation, I was going in a targeted way in directions that I think could be behind the mechanism. And it was 2 to 3 years of frustrating negative results that also help us understand the situation, but do not yield results as we hope. And then we decided to do omics, the large-scale studies, that in one experiment in few days, we get data of so many different metabolites and different lipids that helped us to understand more the situation. So I think there was a bit of delay or in progressing with this study.
Thank you. Dr. Wiese?
Yeah. So I think the biggest barrier that I really came up against in my research was actually in the publication process. And so the paper is published in Frontiers in Neuroscience, but that was after 7 rejections. It was very difficult to get comments back that like people who use drugs aren't going to take steps to keep themselves safe, which as a harm reductionist and someone who's worked in the harm reduction field for longer than I've been in science, I know that that's not true. And so I think battling the bias and stigma that still exists around people who use drugs and people who use opioids was probably the biggest barrier in my research.
I'm really sorry to hear that, but thank you for sharing. I think I may have skipped over you a moment ago. Did you have the chance to talk about how you hope this research will become a part of medicine? What's your hope here?
Yeah, so my hope is or what I would love to see is a way that muffin a way that we can have CBD available and accessible to people who use drugs, the people who need it. Like, I don't want to see it priced out of access. And the hope would be that we can determine the best route of administration as well as the best dosing, where folks could consume CBD alongside of their opioids at the same time to prevent overdose. And then in the event that an overdose were to still take place, like my research suggested, the hope would be that we can use the least amount of naloxone as possible, given that naloxone induces precipitated withdrawal symptoms in people who are dependent on opioids, and that can be a very traumatic experience for someone. So my hope would be that it can help make opioid use safer and increase the safety window profile of opioid therapies for more people to be able to um, enjoy the benefits of opioids with less of the negative side effects.
Okay, thank you. And do you see an opportunity for this to translate into accidental overdose prevention for people who are not addicted to drugs but are taking pharmaceuticals, opiates for pain management?
Absolutely. Just like I think there's been a big push, a move to every time an opioid is prescribed, naloxone is also prescribed, even though you don't intend on someone overdosing on their pain medication. It's a safety practice to have naloxone in the home for anyone who's utilizing pain management through opioid therapies. And the hope would be that the route of the formulation of CBD that's found to be most effective could also be prescribed at the same time as the opioids. And so folks at home who are, even if it's just like an acute injury that they're taking opioids for, could also take CBD alongside as hopeful preventative to an accidental overdose even taking place.
That's great. All right, so we have a question here. How long would it take breathing rate to return to normal after hydrocodone without the CBD?
So the furthest out that I looked at the breathing behavior was only about I believe it was like 45 minutes. But I did not follow it out far enough to see when it returned to baseline. I can say that the baseline recordings that I collected were at least 6 hours post the last dose. Um,
Okay, great. Well, Ethan, do you have a question for either of our esteemed researchers?
Sure. Well, we've heard a little bit about Beth's plans for her discovery. Radka, I'd like to ask you the same thing. What's next for you? I know that you're about to finish your PhD formally based on this elegant work, but what's next?
So that's a great question. First, I need to finish my PhD thesis. I'm almost there. And then I think I will take a little break in my scientific career and think about what I want to do because there are so many options and the world is open, so it's very hard to decide.
Right, excellent. We did have a question if somebody could have contacts for the presenters. I will say that both of them are on LinkedIn, so feel free to connect with them there. Someone did ask, did your staff person report any better or worse pain? So that will be our final question.
So yeah, I was hoping that she would have said, "Oh yeah, I feel great." but she did not necessarily report any changes from the hydrocodone. The hydrocodone managed her pain, and the CBD didn't. Like I don't want to say the CBD didn't have any effect because that wouldn't be a fair statement to make, but she had already managed her pain with the hydrocodone. And so she didn't feel, um, yeah, that is a really good question. But she had already managed her pain with the hydrocodone. So yeah.
Great. Thank you. So we will send out a link to this recording, and we'll include links to the papers on both of these topics. And I just want to give each of you an opportunity to say something about the importance of the community supporting this kind of research. I think money is really challenging to come by. And since this is a fundraising webinar for the ICRS Ethan Russo Trainee Prize, I want to end today with just a little bit from each of you about how important it is for young researchers such as you to have access to money to support your work. So, Radka, why don't you start? And I know you addressed it somewhat at the end of your presentation, but if you could elaborate, because I know it's not easy.
I'm sorry, I had troubles here. And now can you repeat the question?
Yes, of course. So can you talk a little bit, just kind of a final word about why it is so important that the community support financially this kind of research?
Yeah, of course. Yeah, because me as a PhD student, um, I don't have many opportunities to really get money to support my research. It really depends on the background on the laboratory you're at. I know there's variability in that. But if, let's say, if you're less lucky and you don't get as good funding, so even though you have amazing abilities and amazing ideas, without money, you cannot really progress. So prizes like that really help people in my position, at least, to turn these ideas into like real results and real life impact.
Thank you. Beth?
Yeah. Yeah, the award was I was incredibly honored to receive such an award. Um, my research is a little outside of the box in the way that I am looking to expand the safety profile of opioid therapies and prevent overdose before it happens without stopping anyone from consuming the substances that bring them the benefits that they feel they need. And especially that's not always funded, through bigger funding mechanisms, especially right now in the US, the federal funding has been just overhauled, and even community funding for translational work in the streets. So even funding to donate naloxone is disappearing. And so if we don't have ways to support up and coming scientists who have worked in the field and who have lived experience and who are going to the bench to try to answer these questions or to develop interventions that have never been thought of before, it can be really hard to secure the support that they need. And awards like these not only help support the researchers, but it also is a validation and a confidence booster for early career scientists to keep going and to keep pushing the limits and to keep asking the questions and not just, stopping where the funding stops. And so when community comes together and supports the work, then it's the community also who benefits from that work.
Thank you so much. Well, with that, I thank both of you for being here. I also want to thank Chris Duvall, our educational partner at MM411, for her tremendous support this year in raising funds for the prize. We wouldn't have been here this year without her. I also want to say special thanks to Julie Ermish, who has also been a tremendous support to her, and we would not be here without her as well. And to Ethan Russo, we would not be here without you, and I want to give you the final word. And thank you to all of you who donated. Go ahead.
I can hardly improve on the inspiring reports from our two prize winners. Anyway, I hope that people who have attended today find this work as inspiring as we have. And we look forward to future prize winners, and hopefully there'll be many more with your support. Thank you for attending.